Have to adjust 10 things to get started. So I took the liberty of adjusting some of my content during the previous presentation, and again acknowledgements to the AHA and our funding sources. So I was going to highlight the value of EGMs, and as a prelude to addressing my topic, I thought it would be useful to have a couple of background slides and concepts that may be of interest to the audience. So we owe a lot to this gentleman for pathophysiology of arrhythmias. We briefly alluded to his work yesterday, and this is of course a citation classic paper by Mines, and this is the example of how he described re-entry using turtle hearts, looking at the circuit of re-entry seen over here. Now, classic anatomic re-entry is the reason why VT ablation became feasible, and indeed many of our ablation strategies is centered around identification of scars, as we heard in the previous presentation, and trying to, in your mind, when you look at a ECG of a patient with VT who you think has a scar-based arrhythmia, that is the central concept that goes through how you read an ECG, how you look at images, and how you map within the case to identify the footprint of a scar using electrograms. And then of course you try to ablate those abnormal areas. When you're in tachycardia, you can identify a circuit if the patient is hemodynamically stable. If they are not stable, that's where we use all these findings of late potentials, lavas. All these are terminologies, but all these identify areas of abnormal electrical propagation. Scars are very complex. This is a paper that was published by one of our good colleagues. I'm sorry, the most bottom part of it is getting cut out. It's for those of you interested in the reference, this is 2012 Circulation Research. And this is from Bruce Mail's group in Auckland. So here is an example of an experimentally created infarct where they do high resolution confocal imaging. I'll show you a higher resolution of the scar. This is the scar, and if you look at it, it's incredibly complex. Today we're going to be seeing VT cases, and what is happening in these patients with these types of scars is the fact that this is normal myocardium, this is scar, and if you look within the scar, there's actually surviving myocytes and islands. So these are electrical conducting pathways. They lie within the scar. We use the term slow conduction. Electrical impulse does not conduct slowly. It just has to go through a longer distance. The speed of propagation is the same. So this zigzag long propagation sets the stage for re-entry. So the sort of the iconic image that using all the studies that have come from so many labs around the world, we put this together just to explain in the big picture what we are looking at. So scar-based re-entry. Here are just 2 beats of ventricular tachycardia. Here is a histologic scar that we have put together. This is an experimentally created infarct. But these electrograms here are simulated, which is they are from actual VT cases, but this is for illustrative purposes alone. So if you look at 2 beats of VT, and if you look at what happens between the 2 beats, the electrical impulse propagates here. It travels a much longer distance through the scar, and these areas, if you had a catheter that happened to sit at all these sites, it's not always possible because some of these circuits are intramyocardial, but if you can identify critical parts of the circuit, you will see these areas numbered 1, 2, 3, 4, and 5 going through the scar, and that sets the stage for re-entrant ventricular tachycardia. If you're hemodynamically stable, we use all these principles. You're going to see this in the cases today, and all of you do this in your practice. Use entrainment and other pacing maneuvers to identify a scar circuit, a VT circuits, and of course at the right place when you turn on RF. This is among the happiest things you'll ever see. This is an actual case. We're ablating here and VT terminates. So the central concept is scar and ventricular arrhythmias, and what we have learned over the past 15 years, and we owe a lot to Dr. Callens and Marchlinsky and their team at UPenn for helping us even look at electroanatomic maps, to even come up with the simple criteria to define. You need a starting point of what is a scar and what is not a scar. So this is deep in myocardial scars, areas less than 0.5 millivolts, But as it turns out, these are areas that are going to have small areas of viable islands and viable circuits of arrhythmias, and these are the circuits that sustain monomorphic beat reentry. We also know, based on a whole lot of other studies, that the junction between scar and relatively normal myocardium is a complex zone. It's electrically complex, it's structurally complex, and also it has complexity in functional control because of nerve sprouting. And these are the areas it's not uncommon for you to see focal PVCs that actually cause VF. And almost all major VT ablation programs have identified these PVCs, and they've used this to target VF. And it's one of the most pleasing ablations that you can do, because that sometimes presents itself as VT storms. So constantly you're going to be doing this imaging scar, correlation in your mind. So experimentally, one interesting study, this is one of my favorite studies from many years in our lab. Shiro Nakahara, who's now chief of electrophysiology in Tokoyo in Japan, spent 3 years with us, and he had this nice story that was a featured cover story in the journal Heart Rhythm. And the importance of this particular study was the fact that everyone was publishing papers on MRI, including us, identifying scars, but we couldn't quite tell what the electrophysiologic meaning of that scar was. And so Shiro created an infarct, and this is actually a beating heart MRI superimposed on a model. This is a 3D data set. And what you see over here is areas that are outlined here in black are transmural portions of a septal scar. This is an LAD territory scar. And this is high density epi-endo mapping, and it really doesn't matter which system you use, Navex or Carto. They're comparable. Carto is the best validated technology, which when it started off, it was actually a study done by Dr. Callens. And we subsequently validated using Navex, so they're very comparable in terms of their reporting, whether it's electrofield or magnetic-based navigation. And what's interesting is in this particular animal model, closely mimics human infarct. If you sample electrograms from each one of these areas, normal to border zones, and especially the diseased areas, you actually see areas of slow electrical propagation. If you look at this, you have a far field signal, and the near field signal is actually late. So this arrives late because it has traveled a much longer distance to get there. So this is impulses meandering through the scar. And this concept is very, very important. You can see split potentials, late potentials, and of course there are times you can see continuous activation. These are very, very important. And this actually has a lot of value for us, because the next session you're going to be hearing strategies of ablation that Dr. Callens and Natalie are going to be presenting. And the way this works is, and what has been magical for the field of VT ablation is that you can ablate VT in sinus rhythm. You can identify abnormal electrical propagation areas and tackle it. So what is coming down the pipeline is going to be very, very interesting. This is preliminary data that's not yet published. High density maps can be correlated with high resolution MRIs. So you can now start making three-dimensional correlations between scar, geometry, and architecture and electrograms. For instance, this is preliminary data. This is not yet published, but it's important for the group here, since your team here is very interested in wideband and other techniques. It's now realistic because you can actually image patients with implanted devices. And in this case, you can see the complexity of scars is represented in the electrical signal. So these are the types of studies that are in the pipeline, and hopefully offline I can show you more quantitative data on these kinds of image EGM correlations. So let's try to put all this experimental data in clinical context. So when a patient comes to us, is referred to us with VT, how do we approach this? So the clinical approach starts off, and we owe a lot to this gentleman who was a Nobel laureate, and of course 100 years later we really have not made any great advances more than what he had contributed. And it's still the power of this test, which is a testament to a great life and how useful it is. Using various techniques now, we have very, very good algorithms for identifying where circuits are. One recent advance worth highlighting is this work by Yoram Rudy's group, and there's a lot of attention these days for ECGI. ECGI is very interesting and exciting because it has one advantage over traditional 12-lead ECGs, and that is it takes the imaging of a patient, CT or MRI, of how the heart sits in the chest, and has a lot more surface leads, and you can now essentially generate a map which experienced electrophysiologists can just do that in their mind based on experience. So this makes ECG more user-friendly for its display. There are lots of scientific limitations, and we now know that the recent paper published in Circ EP by Bruce Smale's group, the data that I showed, actually shows that there are major error factors involved in ECGI. A simpler version of ECGI is a work that was done by Peter Van Dam, who's one of our colleagues, who uses simple 12-lead ECG, takes a stereoscopic image of ECG's leads on the chest wall, integrates this information, and you can essentially get a projection map. And this paper was recently presented at Heart Rhythm, and the manuscript is under review. So you can actually get a 3D model and identify where a VT circuit originates. So that's clinical ECG. Imaging, already this was shown by Dr. Jimenez about wideband MRI, and this protocol now has been shared with many groups around the world, and it can be performed safely in any device that's implanted ever since 2002, and the artifacts due to MRI can be removed. And this has been one of the nicest things for us, because the most valuable information is in patients who have implanted devices, and getting this type of information is going to lead to new studies. And of course you can integrate wideband MRI images with the electroanatomic map so that you can get structure-function correlations. So using that, let's now address what we would see in the EP lab. So a protocol for how substrate-based activation mapping strategies for VT is shown on this slide. And I was toying with the idea of how to stay within the timeline given by the conference chair, and hopefully we can use this as a way to navigate through a case. Based on a work done by a whole lot of labs, and of course this work again was started off by the group at UPenn, is the central importance of understanding the roadmap of the heart, and that is an electroanatomic map. So once you have that information, that really guides you as to how you do a procedure. Typically when a patient comes into the lab, the first thing we would do is we would do a NIPS, and if the patient does not have an ICD in place, the first thing we do is do a quick EP study so that we understand what sort of VT can be induced. And the patients essentially fall into three groups. Patients who have stable VT, there are some patients who are non-inducible simply because of sedation or anesthesia, and then you have a group that has unstable VT. So if you have stable VT, thanks to so much fantastic work done by Dr. Stevenson and so many groups around the world, we actually have very good strategies of handling this. We can do entrainment mapping, we can map fractionated diastolic activity, we can also do something that's recent, which has fascinated our group quite a bit, and that is moving a catheter. I mean, it's not a dramatically new idea, it's just revisiting a concept that has been known since the 80s, which is if you move your catheter closer to the site of a scar and you pace within the scar, you can actually get the VT started because you're much closer to the circuit, and you can induce areas of unidirectional block to start VT. Once you identify using these maneuvers, you can identify exits, isthmuses, and of course you can ablate, and the hope here is to terminate VT. But having said this, at one time before electroanatomic mapping, that's where the study would stop. After you get the index VT, and if whatever is mappable was ablated. Now that is just one part of the study. Once that is done, we still address the substrate. More or less in some way, shape or form, most labs in the world do this. So unlike the world of AFib ablation, where there are lots of variables and variations on the theme of what people do, there is actually convergence in the world of ventricular tachycardia management and ablation, which is the substrate is addressed. What do we do with the substrate? And the substrate becomes very important if the patient is non-inducible or has unstable VT. The most important strategy is tagging abnormal electrograms, and that's where quality of signals matter, careful attention to detail matters. And once you see this, you pretty much have two major groups. You have patients who have late potentials or abnormal potentials. And once you see them, you can go ahead and use them to ablate, and I'll be showing you a couple of examples of how we do that. The other problem is, and this is not an insignificant group of patients, and I want to really highlight this because our group is quite excited about it, and several other groups who actually see these late potentials highlight a lot about it. But I want you to keep in mind that there are a substantial number of patients, and especially patients with NICM, who have a paucity of late potentials. So we, in all honesty, don't have a good understanding of what we should do with those scars. So this is where several approaches come in: encircling the border zone, encircling the entire scar. These are strategies that people have used, sometimes encircling the scar and trying to connect it to another anatomic obstacle. All these approaches are being used. We don't have a good understanding, and that may be a good point for discussion, and I'd be very interested in hearing the audience view and the other professors' view on what they would do with these cases. So the story begins with high density mapping, and I'd still like to put this up because this is almost a decade old, and at that time this is what used to be very high density map. This is a 357 point map of a large inferior wall scar. As you can see, this is the kind of a map that we would do. It would take 2.5 hours to do this part of the study itself, and of course this used to crash the computer because it couldn't handle that many points. And we've come a long way since then. We now can routinely obtain 1,500 points to get good definition of substrates. And the future iteration of other systems that are coming in are likely to give us more data. But we're also soon going to learn the fact that there is going to be a trade-off between what my son has now taught us. The word is TMI, too much information. So this is the lingo kids use nowadays in texting. So that is a barrier that we're going to face in EP, which is TMI. So what has been the other useful advances? Epicardial mapping has actually added to VT ablation, because you're not just dealing with endocardium, you have another surface of the heart that can be mapped. And of course, standing in the continent we are standing in, I'd like to highlight the fact that when we were asked to do this review, and we also did a whole issue for Dr. Natali's journal, and that is we dedicated this review article to the group in Sao Paulo, Brazil, because it really highlights what I'm seeing again after this is 15 years later that I'm back in South America, and truly the generosity of the people is very obvious. And again, I'd like to highlight and thank them for the fact that they trained at that time. The first two groups who went there were Dr. Natali. A month later, I was a young assistant professor at that time, and they have really gone on and helped so many teams in the world, hosted them. And we were able to actually use this, and we were able to train so many other teams in this, and it's nice to see that it now has a standard place in electrophysiology. So many people have published institutional experiences. We recently put together our institutional experience in this, using epicardial mapping. Now, the purpose of doing all this is to learn science and to try to understand from a limited number of cases. So what is the purpose of academic institutions and leading institution like yours over here? We are here to try to understand science and try to interpret that in a way so that it's useful for the rest of the practice community, and also anything that's published either has to be confirmed, extended, or refuted. And one of our interesting studies, and we were very inspired by studies done by multiple other groups, University of Pennsylvania, University of Michigan, Brigham and Women's, Dr. Stevenson's group, and we really started focusing on electrotrams. So here's an example of late potentials, moderate and very late potentials that we identified based on moving a catheter around various parts of the heart and correlating that with various indices of index activation of the QRS complex. The data is quite intense, and the manuscript, and for those of you who are interested, can look into this. There's one table in that particular manuscript, took about 6 months to develop because it has an enormous amount of metrics on scar dimensions, and some of which reassuringly has stood the test of time, and others we are learning may be affected by things like wavefront of propagation. So that is going to be very interesting. So when you look at this type of data set, when people are showing that to you, in the back of your mind, ask yourself, this is one point in time. It's quite likely in the future, if we can acquire more data, we may be able to bring out late potentials by changing how we activate the scar, bi-V pacing versus RV pacing versus sinus rhythm. So these are all different ways of electrical propagation or entry into a scar. Here is data sets again showing you complexity of EGMs from the endo and epicardium in patients referred for VT. And this shows again how it takes a lot of time. You have to set a good sort of noise threshold in your lab to do this. These are very small potentials, 0.3 millivolts, 0.27 millivolts, and many a times we set the thresholds. If you look carefully over here, the bottom part of the panel is the U-pen settings, 0.5 to 1.5 millivolts. What you see over here is a display between 0.05 to 0.5. This is 50 to 500 microvolts. So when you look at that, it's almost like a Google Earth display. You look at it with one band, and really what you're doing over here is all what is gray over here actually has information that is contained in it. And now we actually have the density of mapping to do this. This took a huge amount of time when it was done. This was 7 years ago, and we're hoping that this is where computing and the core EGM labs that we have set up, in fact one of the big trials, the STAR VT trial, has a electrogram core lab that has been built at UCLA, and our exclusive mission is going to be to help other teams in the world really to understand EGM characteristics, because scientifically that interests us a lot. Why? The data is shown over here. So this is one heavy-duty slide that I'm taking the liberty to show to this audience, because there are differences between non-ischemic and ischemic cardiomyopathy. All of us in various levels have noticed this in our own practices. So here's an example of NICM and ICM. This is quantified data, and again, this is a total of 16 patients with non-ischemic and 17 with ischemic cardiomyopathy. It's a huge amount of data set. So you can imagine there are 2 surfaces to be mapped in all these. So if you look through this, you can multiply both by 2, and that is the number of electroanatomic maps to look at. And if you carefully bin where you see late potentials, this is a very important part of the slide, and that is, you look at the voltage over here and look at the distribution of late potentials. Late potentials live in areas of very low voltage. In fact, if you use the University of Pennsylvania cutoff, which is the 0.5 millivolts, bulk of late potentials are seen within what you would see, or just use the loosely used term scar. But what they identify is 0.5 is the area below which you're going to see a lot of abnormal electrograms. So this dotted line over here shows you that most of the interesting stuff occurs in this area. And a quick look at this image also tells you that there are a lot more late potentials in ischemic cardiomyopathy compared to non-ischemic cardiomyopathy across the board. And if you were to look at this even more carefully, there are big differences in the quality of scar and the density of late potentials. So this is scar area late potentials, ischemic, non-ischemic. In fact, this is the one slide that shows 2 years worth of data. And it's immediately obvious that there are big differences between ischemic and non-ischemic cardiomyopathy. Typically, the scar area is much greater in endocardium and ischemic cardiomyopathy, which is well-known studies. It's been highlighted by Dr. Josephson's group when he was at UPenn, and subsequently has been really standardized by subsequent studies from Dr. Marchlinsky's lab, from Bordeaux in France, from Dr. Natali's group and our own group, showing that this is in general true. NICM, on the other hand, does not follow any of these rules. As it turns out, their scars can be in any location, mid-myocardial, epicardial, sometimes epicardial only, and we still don't quite understand what the pathophysiology of it is. But what we do know is the electrical footprint of the scar is different. Typically, post-infarct scars have more late potentials, have more abnormal EGMs to track. NICM doesn't always read that rule book. which brings up challenges for catheter ablation. So flying through some good examples, here's a study. This was a collaborative study that we did with Dr. Delabella's group. This is multi-electrode mapping, double transeptal. We put in multi-electrode catheters, and of course deep within the scar you can get pace maps, and you can identify these abnormal EGMs that you can tag and ablate. Further, you can also do mechanistic studies. You find scars that have a lot of late potential. So this is a nice study that Rod Tung did when he joined our group, and this was his first project in his fellowship, and it was a series of 3 papers. The first paper shows that if you're spacing within the scar, you can identify multiple exits. So here we are just pacing in the scar. Watch what happens. We are pacing, boom, there is a change. So you can see that the stim to QRS changes and the morphology of the QRS changes. So this is mechanistically very beautiful, because what it tells you is that you can be pacing within a scar and you can actually have multiple exits. Now, it's very interesting to note this. We know that these tend to be arrhythmogenic scars, but there's another point I want you to find a way, and we still haven't quantified and published this. Not all pace maps and not all morphologies that you get from pacing within a scar are correlated with actual clinical VTs. As it turns out, there's a lot of functional control to these circuits. And what really fascinates us is if you have a series of pace morphologies within a scar, there's only a smallest number that result in sustained ventricular tachycardia. So that is pretty fascinating if you think about it. So there are a lot more roads that you can take, but only a handful of roads are available for a VT to sustain itself. Now, this concept can be further extended, and Rod spent a lot of time in a series of cases, and I believe once Dave was even visiting our lab when we were collecting some of these data sets using multi-electrode catheters. What you see over here is a duodecapolar catheter that's been placed, and this is a late potential map. All right. So what you see over here in blue is late, what is red is early. And this is the EGM, these are late potentials. Sometimes, much like how a river flows, if you can go more upstream to the point from which a scar is activated, you may be able to get rid of a lot of late potentials with a few targeted ablations, which is shown right here. Post-ablation, you look at this. These were the lesions that have been placed, essentially a series of just 4 ablative lesions, and all these late potentials have disappeared. So this pretty much tells you there's upstream and downstream to how activation occurs. So how an impulse enters a scar may occur through preferential channels. Again, that doesn't always happen, but when you see it, it's remarkable and it's a lot of fun because it abbreviates your procedure. Otherwise, you have no other option but to extensively ablate. But the world continues to be complex. This again is data from UPenn showing you that we are now dealing with trickier substrates, intramyocardial substrates. But we are electrophysiologists. We don't take no for an answer. Our mission is to search and destroy and control VT. Here's one example where we actually used a neurointerventional procedure. We actually had the neurointerventionist scrubbing with us, and we placed wires within the coronary arteries, and you can actually coil embolize these regions. You can record through the intracoronary wire, you can get an EGM. And alcohol, of course, has uncontrolled destruction, whereas neurointerventional coils give very predictable infarcts that can be controlled. Of course, being in a place like Asia or South America, you still see a lot of valvular disease, and we understand VT mechanisms. Here's an example of a patient who had 2 mechanical valves, had a VT, and you can actually enter this ventricle not using surface approach or hybrid, but transventricular septal puncture. This is through the IJ, this is going through the ventricular septum, we're in the LV, and of course, here it's again the joy of VT ablation. You can identify the circuit, map it, and ablate. So we are non-stop in interventions as electrophysiologists because VT ablation, based on the recent international ventricular ablation collaborative trial, shows a mortality benefit, and hopefully during the discussion session We'll put up 1 or 2 slides just to highlight the new data that's come out. Retrospective, the largest series to date of 2,000 patients, which combines data from 12 centers around the world, showing that there's benefit in terms of mortality based on this retrospective study for VT ablation. So the time that we spend is well worth it. So beyond the heart, of course, is going to be the focus of the lunch session. Sometimes we can't fix the problem at the level of the heart itself, and that's where neuromodulation has been used. And we'll be talking about various types of neuromodulation in the lunch session. So this is just a trailer for that. Higher levels, cardiac sympathectomy, renal denervation. So I'll stop here. We love cardiac interventions, and I'm looking forward to the live cases. I'll stop here, thank my colleagues, and stay on time. Thanks.
I have a quick question. First of all, thank you for your amazing talk. What do you think about the usefulness of the bipolar voltage map to predict the transmurality and distribution of scar?
That's a fantastic question. So there is, pretty exciting data that has been presented by Dr. Callan's group actually on using actually unipolar display for predicting presence of a scar, usually endocardial voltage to predict the presence of epicardial scar. And as it turns out that it actually does play a role, but we think that it requires a certain set of circumstances to make it happen. Bipolar voltage, interestingly, provides a lot more local information. So we've been impressed by the fact that bipolar voltage does not provide that level of correlation, whereas unipolar has a better likelihood, if you will, of showing scar. Now, the areas where that EGM-based method tends to, I wouldn't say fail, but where it becomes inaccurate is if a scar is surrounded by a lot of live myocardium, based on the patchiness of the scar, remote myocardium tends to influence the recordings quite a bit. So it's almost lateral information is what makes the predictability somewhat limited. But I have a feeling that now with more imaging studies that are coming up, we'll be able to use this very effectively. And I think it has a big implication also for things like ECGI, because ECGI highly depends on the projection from the surface as to what happens on the epicardial surface of the heart, because that's how the inverse solution works. So I think those studies, once it's more widely available, will really help us place that data in context. And you can actually combine information from endocardial voltage maps and what you obtain on the surface. And I think that will make the resolution of this data a lot sharper. Do you have a comment on that, David? That's data from UPenn.
I think that was very well summarized. Can't answer it.
One last thing to add, that the problem with unipolar mapping, to me, is that when you face a very prominent endocardial scar, maybe the unipolar map can fool you in terms of the information about the transmurality and the actual distribution of scar. So
actually, I concur with you. I was talking about endocardial voltage predicting epicardial scar, but epicardial scar predicting endocardial voltage absolutely is affected by what you say. And that was not what UPenn published. UPenn used endocardial voltage to predict epicardial scar.
And then there's this whole concept of penumbra,
yes,
which I struggle with. I think what might be the resolution of all of this is that electrograms have limitations and benefits, and imaging has limitations and benefits. And if we can better understand the specific circumstances where one is better than the other to decide which should rule that case, I think that would help.
Absolutely. Our big problem is, of course, the third dimension with EGMs. And it takes a little more effort, and we do it only in a handful of cases where we get into a coronary vessel to get intramural data. That's precisely what happens. If you have a lot of healthy myocardium that surrounds diseased myocardium, that's when most of these fail. And in fact, even imaging would fail because nobody in the world of MRI can agree on what is a gray zone. And we saw some of the data that Dr. Jimenez showed on virtual EP study. That is completely based on gray zone measurement, which Tim Dickfeld, I think, is putting together a review on this. There are 4 potential ways in which a gray zone can be measured. So, as I say, the gray zone continues to be gray. So therein lies the problem, where you have to use all pieces of information to give the best possible outcome for the patient.
And I shouldn't add this, but I can't help myself. I think we blind ourselves to a certain extent, and Shiv brought some of this up when we talk about dense scar. Dense scar has voltage channels through it. That's very well known histologically. Same mistakes are made, I think, just in kind of constricting our further thought when we talk about border zone, or particularly when we try to equate border zone and gray zone. Gray zone is a very difficult topic that might even be a trick of the light.
True.
Although the studies from Hopkins really kind of give it some.
It is. I think the point to highlight, and of course I embarrass David all the time by quoting one of his old papers to him.
Because he understands it better than I do.
No, it's a point is, how it is, right? Many of the papers that we write, when you suffer through it, of course, the reviewers are nasty. They actually are not. Sometimes they look at a data set that we haven't reflected on quite a bit. I think the term border zone will have to be looked at in 2 ways. Border zone is more like, the analogy that we were discussing in our group meeting is, imagine if you're flying in a plane or going in a train, and you're looking outside. Normal myocardium would be a place that has green scenery. You're driving through it, you see rivers, your train is going through it. Then what happens is, let's say you fall asleep and suddenly wake up, you're in a desert with occasional oases. But what is in between the two, if you had stayed awake, the scenery gradually changes. So in other words, the border zone is a zone, and that zone has 2 components. One is actual physical changes, where there is more myocytes, more scar, so there is a trade-off between the 2. But there's also a functional control. So we use the term line of control for this, and a recent study that's almost concluding based on human data, because this is where we put a lot of pressure on the basic scientists, although we do a lot of cellular and molecular studies. Homo sapiens data matters a lot, because Homo sapiens write the checks, and we owe it to them to get some answers. But I think what is a border zone is where the impulse propagation stops. That is a line of control. And that can be sometimes deep within a scar, sometimes it can be very proximal. And that's where scar geometry and border zone geometry matters. Because I've reflected for the longest time, why does border zone ablation work? Dave Wilbur has reported this. Your group has reported this. In fact, remember the spoke model of ablation. I know for a fact that when I'm completely back to the wall, we just go encircle what we define as the border zone. And I think it probably works by preventing entry and exit of impulses. And that's where I think the term, the iconal relationship, which is the wavefront of how an electrical impulse comes and hits the scar, matters. So if you have a very smooth border zone, I think the chances of a wave reaching that is much different compared to a very rocky border zone, if you will. Because look at the heart, right? The sources are small, but the sink is huge. So a small number of myocytes are able to control a lot of myocardium, starting with every minute of our lives, the sinus beat, right? How does the sinus beat actually control the rest of the atrium? And all that is because of preferential propagation, and the scar propagation is where I really think that we're going to learn a lot. And we ought to focus a lot more on EGM research, and that may give us new clues on coming up with more applicable therapies.
This is very interesting, and we should stop, but this border zone idea suffers from the same problem as gray zone does specifically, yes. Or otherwise this wouldn't work. Yep. And I remember being fascinated when I was actually doing more work in the animal lab. We looked at the histology of porcine infarct, and the point's been made that it represents human infarction pretty accurately. But we're really surprised at this lack of border zone. There's infarct normal. So what if our ECG measurements, or electrogram measurements, or border zone are just straddling that oasis versus desert? and the voltage looks greater to us just because it's measuring both. Oh, that's a good point too.
See, but if you look at this, right, this is why later offline we'll discuss this. This is a smooth border zone, and it gives you an electrical potential, in this case bipolar, but we can give you high density data. And we've also done plunge electrodes, whereas look at this border zone. So this is an area where the iconal relationship will allow propagation out of a scar. The most fascinating scientific question is, how does a small sliver of impulse that comes from the scar, right, be placed within a scar, how can it control the rest of the myocardium? Because if you look at this electrical signal, right, it's like 2 or 3 people entering a room. Everyone else there, right, has a bigger say, but that impulse is able to go control the rest of the myocardium. There's a lot of, the wavefront curvature matters a lot because the cells in front of you have to be depolarized. That's how impulses propagate. And this is where I think the complexity of the border zone and the functional control, this can be, I think now based on 10 years of research, I know that this can be done in humans in very controlled setting. Like that's why we're reinvigorating intraoperative mapping, for instance. Again, life is never easy. When you were an intern at UPenn, it was always a challenge to go to the OR, find the time, do people have the protected time to do this, but you're going to see a lot of research coming out in this area, and I think it's worth focusing on, because this is a tractable problem. This is not, out there, pie in the sky type stuff, because it's a common problem. And small scars may kill. And we never should forget the fact that 7 million people die suddenly every year. If all of us in this room for the rest of our lives, day in, day out, worked and did VT ablations, we would not have an impact on 7 million people dying suddenly every year. 10 years later, we'll be at a Latin American VP live meeting and 70 million people would have died. I mean, that's a population of nations if you think of it. That's, for the younger people in the audience who want to be inspired, it's a Holocaust every year. That is how massive the problem of sudden death and ventricular arrhythmias is. So if you needed anything to make you wake up in the morning and think of science and help patients, this would be a good problem to think about.
More to follow. Thank you, Shiv.
Thank you so much.
If we can, really quickly, can we connect